Hello, everyone, and welcome to today's webinar, Mitigating ADC Toxicities with Linker-Payload Design, De-risking downstream ADC development by optimizing preclinical assays with PEG-containing linker payloads. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Vector Laboratories. To learn more, visit www.vectorlabs.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Matthew Giese, Senior Scientist with Vector Laboratories. Matthew, you may now begin your presentation.
Okay, thank you for the introduction. Welcome, everyone. Thank you for attending. Hope you're having a great day. My name is Matt, and this webinar will be about using strategic linker payloads designed to mitigate off-target toxicities of ADCs. The balance between efficacy and tox is a bit of a tug of war, and this webinar is going to focus on tox because many of the potential underlying mechanisms are influenced by the physicochemical properties of the conjugate, and the linker payload can have a substantial impact on those properties. The chemistry group here at Vector makes PEG-based crosslinkers, and this webinar will present examples where ADC developers have used crosslinkers such as these to optimize ADC performance in assays that can suggest downstream tox liabilities. So first, we're going to start out with a recap of some ADC basics. We're then going to present some examples with linear PEG stretchers, and this will be followed by examples with orthogonal PEG modifiers. And then we're going to wrap it up with an overview of Vector's DPEG-based building blocks and crosslinkers that you could use to synthesize conjugates such as these. Okay, so let's go. ADCs are indeed the most successful class of actively targeted drug delivery systems, and this table shows the clinically approved ADCs. There's been a lot of success with passive targeting in drug delivery, and there's been some success using other targeting vectors such as peptides, but using antibodies as the targeting vector has yielded the greatest number of clinically approved ADCs. However, as this table reveals, treatment-related adverse events are not uncommon, and they can result in dose reduction or discontinuation in the clinic. A 2023 meta-analysis found that in the 169 clinical trials conducted since 2000, which involved close to 25,000 patients, the incidence of treatment-related adverse events was 91% for all grade and 46% for grade 3 or greater. Furthermore, the failure rate in the clinic is not insignificant. And of the 97 trials terminated since 2000, 84% were in phase 1 or phase 2. And for the vast majority of these, it was due to lack of efficacy at the maximum tolerated dose or due to safety and tox concerns. So while ADCs have seen some success, there's substantial room for improvement, particularly with respect to tox and safety. Now, the reasons for failure due to tox in the clinic are manifold, but this webinar is going to focus on the underlying mechanisms of targeted drug delivery with antibodies, and particularly the preclinical assays that can be used to suggest something is amiss. So targeted drug delivery is often selective for a tissue and not specific. It's very difficult to thread that needle and get the therapeutic to only one location while avoiding all others. So the goal of targeted drug delivery is to tip that balance and reduce the therapeutic systemic exposure while increasing its exposure at the site of action. However, the reality of targeted drug delivery with antibodies is a little bit different. And while tumor exposure is generally increased, antibodies are taken up and processed in other tissues. For instance, highly perfused tissues like the skin, GI tract, and muscle are very efficient at catabolizing antibodies. In addition, organs with high levels of professional phagocytes, like the liver, spleen, and marrow, they're very good at processing antibodies and their associated immune complexes. In addition, the PK/PD of the free payload must be considered. It is coming off of that delivery vehicle eventually. So all of these factors can contribute to these somewhat common toxicities that are seen across the different ADC platforms. So let's review the underlying mechanisms of ADC internalization and payload release. So first, this is the typical MOA given for an ADC. You have the conjugate, the tumor, and its target on the tumor cell surface. The ADC is administered via IV. It extravasates from circulation, diffuses through the tumor, and reaches its target on the tumor cell surface. After binding, it undergoes internalization and trafficking through the endolysosomal pathway. And during this process, the antibody backbone and cleavable trigger are catabolized. This results in release of either the linker payload or the free payload, and then they can escape the lysosome and engage their intracellular targets and kill the cell in accordance with the cytotoxin's mechanism of action. In the field of antibody therapeutics, this is what's called target-mediated drug disposition, and it's one of the processes responsible for the clearance of the antibody. In the field of ADCs, this is also a process that's responsible for payload release from internalizing antibody-drug conjugates. However, this is an oversimplified picture. Instability in circulation can result in premature payload release or linker-payload deconjugation. This is not a huge deal if these entities diffuse into the tumor. The tumor itself is not a vacuous box, but is packed with vasculature and lymphatic vessels and ECM components. Fortunately for the ADC, the vasculature is aberrant and leaky, and this can facilitate extravasation. However, the densely packed collagen fibers and ECM proteins can make tumor penetration difficult. During residency in the TME, conditions and enzymes can also result in extracellular payload release, and this may or may not be desired. The ADC can bind to the neonatal FC receptor, and this can salvage it from the endolysosomal pathway and recycle it to the cell surface, preventing catabolism and payload release. But this depends on where that trigger and linker are designed to release the payload along the endolysosomal pathway. Recycling and release are kind of competing mechanisms in the different cells. The payload itself can also undergo efflux from the targeted cell, either by passive diffusion or by active PGP-mediated transport, and this can decrease efficacy. Sometimes, because tumors are heterogeneous and they have cells that don't express the targeted antigen. So now that extracellular payload can diffuse into these antigen-negative cells to increase efficacy via the bystander effect. But this is still an oversimplified process because pinocytosis and receptor-mediated endocytosis are also both endogenous internalization mechanisms, and these are antigen-independent. Pinocytosis is a nonspecific fluid phase endocytosis, mostly in the endothelial cells lining the blood vessels, but it's very efficient at catabolizing IgGs, and this can release the payload. Receptor-mediated endocytosis, facilitated by FCGR binding, can trigger internalization and payload release. And the payloads can kill the cells, they can diffuse out, they can diffuse into antigen-negative cells for a bystander effect, and because this is in the tumor, it's all good. But now it's fairly well established that less than 1% of the administered dose reaches the tumor and is processed in this way. So where's the other 99% of it? Well, there are other tissues and organs, and the ADC and the payload can be carried into these other tissues and organs and subjected to these same mechanisms of internalization and processing. Pinocytosis is very efficient in tissues rich in capillary beds with high blood flow. FCGR-mediated RME is hard at work in tissues with high levels of professional phagocytes. In addition, the antigen is sometimes tumor-associated and not tumor-specific, and now it's expressed at low levels in healthy tissues. So now the ADC can bind to that antigen in the healthy tissues, and it gets subjected to internalization and processing, and this can result in either payload release or it can be recycled to the cell surface. So this is presumably the fate of the other 99% of that ADC. And regardless of where the ADC is taken up and processed, the payload has the potential to re-enter circulation and enter other tissues until it is eventually eliminated. So the reason I think it's important to review all this, particularly with respect to the vector linker portfolio, is that these other processes besides TMDD are influenced by the physicochemical properties of the linker payload. And those are influenced by the different properties of how you put it together and how you build your payload. So properties such as size, aggregation state, multimeric state, hydrophilicity, charge, and even spatial arrangement can affect things like extravasation, tumor penetration, cellular internalization, diffusion into and out of cells, interactions with cell membrane surfaces, and interactions with the various FCGRs. And all of these processes have the potential to affect both toxin efficacy depending on where and when they occur. So the goal of ADC design is to get more of the tumor and less of it to the other organs. And this is kind of a tug of war between TMDD and these other undesired non-targeted processes. So I'm not sure this is exactly a zero-sum game, but reducing the amount of the conjugate that is consumed by these other processes makes more available to the tumor and vice versa. So this webinar is going to present examples where developers have used linker payload designs that incorporate a PEG to reduce these undesired processes, or at least improve the performance of the ADC in an assay that can be impacted by these underlying processes. So there will be a number of preclinical assays that will be covered in this webinar that can be used to predict downstream tox liabilities. Excessive hydrophobicity may suggest that nonspecific uptake will be an issue. Instability could suggest reduced delivery of the payload to the tumor or an increased systemic exposure of that free payload. Antigen-independent activity can indicate non-targeted activity in healthy cells. Accelerated clearance of the total ADC relative to the undrugged antibody suggests drugging the antibody has had a detrimental impact on its PK, usually an increase in nonspecific clearance. Accelerated clearance of the intact ADC relative to the total ADC suggests either instability or accelerated clearance of the high DAR species is a problem, or a combination of both. Tolerability, as assessed by a loss of body weight greater than 20%, suggests tox. Less than 10% or 20% might suggest a narrow therapeutic window. It's almost essential to run clinical pathology studies in discovery phase. Antigen-independent activity can indicate a nonspecific uptake by these non-targeted tissues, and again, suggest tox. And biodistribution studies showing accumulation of the ADC or the free payload in non-targeted tissues can suggest toxicity to those non-targeted tissues. So this webinar will present examples where PEG-containing linker payloads have improved assay performance in one or more of these assays. And ideally, the more boxes that can be checked, the lower the risk of insurmountable tox issues later on in clinical development. I wish I could give you a guarantee, but I can't. It's just more about minimizing risk. So there are a number of ADC features that can be optimized to improve efficacy and reduce tox. This can range from the antibody itself to the cytotoxin, to the conjugation chemistry, to the release mechanism, and incorporating a hydrophilic component in the linker, in this case PEG, but there's also PSAR or sulfamides, is one of those important alterations. Due to the combinatorial nature of an ADC, it's often many changes that add up to a better performing conjugate. So while this webinar is going to focus on the PEG component, it's often done in combination with these other changes. And these examples will show the broad applicability of a PEG with different conjugation chemistries, different DARs, different payload classes, etc. This color scheme will be used throughout, and the components in black, purple, and pink will be, that's kind of similar to the products that are sold in the vector linker portfolio. So now we'll present some examples where these linker payloads have improved performance in those assays. And we'll start with some linear linker payload designs. These put the PEG stretcher between the antibody and the payload and are the original incarnation of PEG-based linkers in antibody drug conjugates. So this first slide is a compilation of studies on talarine and tesirine. Recall an overall accelerated clearance or a large delta between the PK parameters of the total antibody and the intact ADC can suggest something is amiss. So talarine is shown on the left here, and this was site-specifically conjugated to 3 different antibodies that give these 3 different ADCs. And these were evaluated in various preclinical and clinical studies. However, in all cases, there was either a large delta or a short half-life and low exposure. And development of all of these ADCs was discontinued at some point. On the right is the redesigned linker payload. This is tesirine, and it uses a different cytotoxin, SG3199, as a self-immolative spacer, and it uses MP-PEG8 for conjugation. This was site-specifically conjugated to 2 different antibodies to give Rova-T and Cami-T that were evaluated in various clinical trials. However, again, in both cases, there is either a large delta or a short half-life and low exposure, and development of both of these was also discontinued at some point. In the studies on the left, it was found that conjugating talarine to the hinge cysteines resulted in significant aggregation, and this could be ameliorated with that site-specific conjugation strategy. However, this redesigned linker payload allowed hinge cysteine conjugation to be revisited. So changing the conjugation site back to the hinge cysteines and targeting a different antigen provided this ADC, which now has a small delta, a long half-life, and a good exposure. And this is Xylanta that was approved in 2021. So this example demonstrates that this optimal linker payload design can be coupled with a conjugation strategy to improve the PK parameters that can sometimes be indicative of off-target toxicities. This is a good example from AstraZeneca showing that the inverse relationship between DAR and clearance can be mitigated with a linker payload that incorporates a PEG-8 stretcher. In this case, the linker payload can be compared to tesirine, how it uses this less potent and less hydrophobic cytotoxin, SG3650. The reduced potency can be compensated for by increased drug loading. And indeed, This linker payload design facilitated the construction of DAR 2, 4, and 8 ADCs with minimal aggregation. In addition, PK studies in rats given equal payload doses found that the PK parameters were similar for all of the ADCs regardless of DAR, and they weren't that much different from the unconjugated antibody. So this suggests that high drug loading did not have a detrimental impact on the PK. When these conjugates were screened at subcurative equal payload doses in xenograft models, they were also equally efficacious. So again, this indicates that the high drug loading did not accelerate that non-specific clearance and decrease efficacy. Finally, tox studies revealed that the maximum tolerated dose in monkey was 5-fold greater than the efficacious dose, and that's compared to 3-fold greater for tesirine. Furthermore, if you note the difference in the PK parameters between the total antibody and the ADC, they suggest linker payload loss. However, the tolerability studies show that the penalty for this is not as severe with these lower potency payloads. So this example demonstrates that this optimal linker payload design with the PEG-8 stretcher can reduce aggregation and accelerated clearance and improve the tolerability. This example from Mabwell shows that payload delivery and tolerability can be improved with a PEG-4 stretcher. And in this case, they use their proprietary dye-substituted maleimide, which gives stable disulfide rebridged DAR4 ADCs after ring opening. The comparator for this study is EV, or PADCEV, shown below. So it should come as no surprise when these two conjugates were evaluated for stability in monkey serum. EV demonstrated significant payload loss, while Mabwell's ADC was much more stable. The internalization studies were interesting, and in high-expressing cells, both ADCs internalized similarly. However, in medium-expressing cells, Mabwell's ADC internalized better than both EV and the unconjugated antibody, and that's despite identical binding affinities. So that suggests that some property of the linker payload or conjugation, some property like the extra negative charge or the ARO ring or the PEG spacer affected either TMDD or antigen-independent uptake. And depending on which, this could suggest increased efficacy in tumors with low antigen expression, or it could suggest increased tox due to antigen-independent uptake. PK studies of both the ADC and the free payload indicated that despite the increased stability, Mabwell's ADC was cleared faster, and its plasma AUC was 20% lower than that of EV. However, it also had a 16% greater delivery of MMNE to the tumor. And I don't show the data, it was also more efficacious in xenograft models. So this is a good example of that duality I was talking about. Up to now, the examples have suggested that increased stability means increased exposure means increased efficacy. But in this case, the opposite is actually true. And a reduced systemic exposure of the ADC appears to indicate greater efficacy. So faster clearance and low exposure per se is not necessarily a bad thing, depending on what is doing the clearing. And in this case, it appears that the linker payload has increased the clearance by the tumor, which is a good thing. So this ADC was also evaluated in monkeys, and the reduced systemic exposure compared to EV was also noted here, and it had a lower mortality rate. Mabwell is currently in clinical trials with this candidate, and when compared to EV, it appears to have a better objective response rate and a lower occurrence of treatment-related adverse events. So it'll be interesting to see how this goes in phase 3. Preclinical studies did show relatively high liver uptake, but Mabwell is using this linker design in their other clinical stage candidates. So this example demonstrates that this PEG-4 linker or PEG-4 containing linker can help improve payload delivery and tolerability and probably increase the therapeutic index. This is a great example of ADC optimization from AstraZeneca, and they show that stability and tox can be improved by a linker payload that incorporates a PEG-8 stretcher and this proprietary topo-1 inhibitor. So all 4 of these ADCs use the same proprietary topo-1 inhibitor, and then for conjugation, they either use MC or MP PEG-8, and the cleavable trigger is either GGFG or Val-ALA. And then they mix and match the various components, and they look at them in these different set of assays. So the least hydrophobic, at least by HIC, is the second from the left. The most stable conjugates are the 2 on the right. So linker payload deconjugation via retromichael and stabilization via ring opening are kind of competing reactions. So you do get some linker payload loss with all of these. However, it's minimized with the MP PEG-8. When evaluated for efficacy, the MP PEG-8 GGFG is the least efficacious, although it's difficult to differentiate the other 2. However, tox studies revealed clear differentiation. And this last example, this last combination with MP-PEG8 Val-ALA showed only mild and reversible hematology changes. So this last one is also AZD8205, which is currently in phase 1 clinical trials. So this is a nice example showing that the different ADC and linker payload components can be evaluated in these different assays to improve physicochemical properties and reduce tox in these preclinical assays that might suggest potential downstream tox issues. This is a good example from CGen showing that stability, PK, and non-targeted uptake can be improved with the linker payload that incorporates a PEG-4 stretcher in this proprietary topo-1 inhibitor. So in this example, conjugates with this linker payload, LB, were compared to conjugates with this linker payload, DT. The conjugates with LB had reduced HIC retention time relative to conjugates with DT. And they also exhibited a significant reduction in linker payload conjugation. Comes as no surprise. When conjugates with this new linker payload were evaluated in PK studies, it was found that the concentration of total antibody was not that much different than the undrugged antibody. So that suggests that this high drug loading did not have a detrimental effect on the PK parameters. On the tox side of things, when conjugates were evaluated in an assay designed to predict nonspecific uptake by the liver, there was clear differentiation. And conjugates with LB were taken up 7 to 10-fold less than conjugates with DT. This low nonspecific uptake was also correlated with favorable rat tox studies. And where rats were dosed with non-targeting conjugates, they had hematology measures that were similar to vehicle, and there was minimal loss in body weight. So this example demonstrates that this linker payload design with the PEG-4 stretcher and the proprietary topo-1 inhibitor can improve physicochemical and pharmacokinetic properties, and then it can also reduce non-targeted uptake and myelosuppression, which might suggest reduced downstream tox liabilities. This final example in this section shows that antigen-independent activity of degrader antibody conjugates can be improved with linker payload designs that incorporate a PEG stretcher. These examples come from different papers, so they degrade different POIs, but they both target HER2, they both use disulfide rebridging for DAR4 ADCs, and they both recruit VHL. So I think we can make some inferences with those caveats, of course. So on the left, both targeting and non-targeting conjugates were prepared, and they were tested for POI degradation in cells expressing HER2. In the example on the top, it can be seen that there is POI degradation with the targeting conjugate. However, in the example on the bottom, there is also POI degradation with a non-targeting control. So the authors suggest that this antigen-independent activity is due to non-specific uptake of that hydrophobic conjugate. In the example on the right, PEG stretchers were incorporated into that linker payload to give a more hydrophilic linker payload design. And then this was tested for POI degradation in cells that both expressed or didn't express the antigen. So in the example on the top, it can be seen that there is POI degradation in the HER2 expressing cells. And then on the bottom, there is no POI degradation in the cells that aren't expressing HER2. So there's no antigen independent activity. So this is a good example showing how these hydrophilic linker payload designs that incorporate PEGs can reduce this antigen independent activity that has the potential to give rise to off-target toxicities. So now we'll switch gears and look at some linker payloads that incorporate these orthogonal PEG modifiers. And these put a PEG in an orthogonal shielding position, so they can not only compensate for hydrophobicity, but they can provide shielding and maybe accommodate different functional groups if desired. This is a good example from AstraZeneca showing that stability and efficacy can be improved by designing a linker payload and conjugation strategy to evade an offending systemic enzyme. 4 different ADCs were prepared with this val-cit trigger and different distances between the antibody and the trigger. In format A, it has no stretcher in the middle. Formats B6 and B24 have PEG6 or PEG24 stretchers. And format C, again, has no stretcher, but it places that PEG in that orthogonal shielding position. The val-cit trigger is susceptible to cleavage by circulating CES1C in mouse, and that was verified here for formats B24. However, CES1C has a very restricted active site access, and that can be leveraged by some of these conjugates. In vivo stability studies found that formats B6 and B24 with that long conjugation distance were extremely unstable. However, format A with the short conjugation distance was much more stable. But now that hydrophobic payload is exposed, and it could present some downstream liabilities with development. Format C, which also retains that short conjugation distance and places the PEG in the orthogonal shielding position, was just as stable as format A. So presumably, this increase in stability is going to reduce toxicity. And it probably comes as no surprise that in xenograft models with circulating CES1C, efficacy also tracked with stability. So while this is a species-dependent phenomenon and not expected to be an issue in monkey or human, it does demonstrate this important concept of using linker design to leverage steric shielding from the antibody and hydrophilic shielding from the PEG somewhat independently, and this can prevent premature payload loss. That same linker payload design was used in a former clinical candidate from OBI Pharma, and they demonstrated that FCGR affinity could be reduced with this linker payload. There's been a couple studies that have shown that conjugating smaller linker payloads to hinge cysteines doesn't have a substantial effect on FCGR binding, but conjugating larger structures like PEGs to that region can. And in this case, it was demonstrated that this linker payload did reduce FCGR affinity. Given that these receptors are expressed on professional phagocytes, this has the potential to reduce uptake by or increase clearance from tissues containing those cells. So while it's only speculation, this might be supported by the biodistribution studies. There was initial distribution to the highly perfused tissues, including the organs of the MPS with the professional phagocytes. However, Cmax was reached after only 4 hours. And after that, there was a rapid decline, and this was accompanied by a slow, prolonged uptake in the tumor. And this resulted in tumor-to-tissue ratios greater than 1 by only 8 to 24 hours post-dose. So while development of this conjugate was discontinued last year, it demonstrates this important concept of using the PEG to shield functional domains on the antibody that might affect these off-target uptake and contribute to downstream tox issues. This is another clinical candidate from OBI Pharma, and they demonstrate that non-targeted white blood cell interactions can be reduced. So they don't disclose the nature of the linker, but it could be similar to the previous example. When evaluated against Dato-DXd, it was found that it had a more favorable safety profile in monkey, and it also had reduced interactions with white blood cells. And this can indicate a lower risk of myelosuppression later on in clinical trials. Given that these white blood cells express all these FCGRs, and the previous slide demonstrated that orthogonal PEG attenuated these FCGR affinities, this could be a similar linker payload design. So both the last example and this one demonstrate the types of studies that can be done to design linkers that minimize these non-targeted interactions that might contribute to potential downstream tox liabilities. This might be a good place to note that some developers like Zymeworks and Eli Lilly and Tubulus are using Fc-silenced antibodies in some of their clinical stage candidates. And this could be another way to attenuate that interaction. We're going to spend a couple slides looking at some examples from Seagen, since they do a really nice job of exploring how these linker variables affect the different assays. In this study, they show that clearance and non-targeted uptake can be reduced with this orthogonal PEG modifier. So they made 3 different ADCs. In ADC 6, it has the MC group for conjugation and an orthogonal PEG modifier. In ADC 4, it has the MC group for conjugation. And in ADC 5, it is the MC group and puts a linear PEG stretcher between MC and that linker payload. When these ADCs were evaluated in PK studies, it was found that ADC 4 was cleared quite quickly. And this is not uncommon for DAR8 ADCs with this MMAE payload. ADC 5 with the linear PEG stretcher was cleared even faster. However, ADC 6 with that orthogonal PEG modifier was cleared much slower. And in vivo efficacy also tracked with clearance. Conjugating MC to hinge cysteines is notorious for linker payload deconjugation, so that was explored as a possible contributing factor. However, evaluating these ADCs in rat plasma revealed that they had identical rates of linker payload deconjugation, so that wasn't the difference here. However, when Sprague-Dawley rats were administered a 6 mpk dose, IHC revealed significant differences in hepatic uptake. And the faster clearing ADC showed the darkest staining, while the slowest clearing ADC showed little to no staining. So this example demonstrates that this orthogonal PEG modifier can reduce that accelerated nonspecific clearance and reduce nonspecific uptake by the kidneys, by the liver, I'm sorry. In a follow-up study, it was demonstrated that efficacy, exposure, and tolerability are affected by that length of that orthogonal PEG. In this study, they used the self-stabilizing MDPR group to prevent linker payload loss, and then they varied the length of that orthogonal peg. These ADCs all had nearly identical in vitro cytotoxicity, but they showed a clear differentiation in these xenograft models. And those with the PEG 8, 12, and 24 show the greatest amounts of TGI, and PEG 8 and 12 appear to be slightly superior. This trend was reflected in the PK studies, and conjugates with the 8, 12, and 24 had PK profiles that were similar to vehicle. This was also reflected in tolerability studies, and the ADCs with the 8, 12, and 24 were tolerated similar to vehicle, with the PEG 12 appearing to be slightly similar. So this is a nice example showing that orthogonal PEG modifier can be optimized to provide this best balance of efficacy, PK, and tolerability. In another follow-up study, digging into the mechanisms of what's going on here, it was demonstrated that non-targeted uptake by Kupffer cells, which are the resident macrophages in the liver, also depend on the length of that orthogonal PEG. In this study, non-targeting conjugates were administered to the rats, and the length of that orthogonal PEG was very it was an in vitro assay, it wasn't administered to rats. But it was found that this non-specific uptake depended on the length of that orthogonal PEG. And ADCs with the PEG 12 and 8 were taken up to the same extent as the undrugged antibody, while those with no PEG or short PEG saw a 2 to 3-fold increase in non-specific uptake. So ADCs with FC engineered regions were also used, so they didn't have FCGR affinity, and they behave similarly. So this does not appear to be primarily a FCGR-driven process, at least in Kupffer cells. But this study demonstrates that the orthogonal PEG can alter this underlying mechanism of antigen-independent uptake, whatever it might be. In a follow-up study, it was also found that the payload concentration in non-targeted tissues depended on the length of that orthogonal PEG. In this study, non-tumor-bearing rats were administered non-targeting conjugates, and the concentration of free MMAE in the liver, spleen, marrow, and plasma was determined by LCMS. It was found that ADCs with the longer PEGs resulted in payload concentrations that had a nearly 2-fold reduction in Cmax in these non-targeted tissues relative to the ADCs with the shorter PEGs or no PEG. All the ADCs also exhibited difference in Tmax, and it occurred at about 6 hours in the liver and 24 to 48 hours in the spleen, bone marrow, and plasma. So in this case, it was suggested by the authors that this rapid non-specific uptake by the liver resulted in catabolism of the ADC and release of the free payload, which slowly diffused into these other tissues. So this study shows that this orthogonal PEG can be modified to reduce this non-specific uptake and catabolism of the ADC in this undesired tissue, the liver. And then this can also decrease the concentration of the free payload in the off-target tissues in the spleen, bone marrow. That same study demonstrated that the tolerability and clinical chemistry parameters depended on the length of that orthogonal PEG. And ADCs with the no PEG or short PEG showed very low survival rates, while those with the 8, 12, or 24, the animals showed 100% survival rates. It was also found that hematology and serum chemistry depended on that PEG length, and while all the ADCs showed some neutropenia, those with the longer PEGs showed less myelosuppression and liver enzyme elevation. So this is a nice set of studies in these last slides showing examples of how the orthogonal PEG can modulate these underlying mechanisms of non-specific uptake that can contribute to dose-limiting toxicities later on in clinical development. This idea of using the orthogonal PEG to shield undesirable physicochemical attributes is also being adopted for other payload types. And in this example, it is a study from Tubulus showing that stability in PK of DAR8 EXA ADCs can be improved with these orthogonal PEG modifiers. So during the initial development of ADCs with topo-1 inhibitors, it was found that EXA could not be used as a payload because it resulted in aggregation at DAR8. So DXD was developed, and this facilitated the construction of DAR8 ADCs. So that's in HER2, which is shown on the top here. This is the control in these experiments. Tubulus was able to use their proprietary conjugation chemistry and these orthogonal PEGs that are capped with hydroxy terminus, and they were able to make these DAR8 EXA ADCs with the val-cit trigger in high yield and little aggregation. And this is TLP3 until TLP5, shown on the left there. When TLP5 was compared to Enhertu in PK studies, it was found that total antibody concentrations were similar, but it should come as no surprise there was significant deconjugation with Enhertu, but TLP5 remained quite stable. Evaluating the PEG lengths, it was found that the PEG-24 provided greater physical stability, and it also provided greater exposure. So TLP5 was compared to Enhertu in xenograft models, and it was also shown to be much more efficacious. Tubulis currently has 2 clinical candidates with this linker payload, and they've also published similar studies using MME as the warhead. So this example demonstrates that that orthogonal PEG modifier can be used to improve physicochemical and pharmacokinetic properties that might suggest improved tox profiles later on in clinical development. And in this case, it appears that it's the PEG-24 hydroxy that is most optimal. In the CGen case, it was the PEG-12 methoxy. So to wrap up this section, well, there's an example showing that the antigen-independent activity of immunostimulant conjugates can be improved with these linker payload designs. And this is a study by Mersana. In the conjugate on the left, a linear PEG stretcher is incorporated between the antibody and the payload, and it's non-cleavable. In the example on the middle, there's an orthogonal PEG that's capped with these very hydrophilic sugar moieties, and it has a cleavable dipeptide. And the example on the right, it has that same orthogonal PEG modifier with the sugar residues, and the cleavable dipeptide has been replaced with a cleavable ester and a PEG stretcher. And then these 3 linker payloads are conjugated to both targeting and non-targeting antibodies to look for both antigen-dependent and antigen-independent activity. So when the targeting conjugates were tested for activity, they all displayed equal efficacy. So they did not differentiate on the level of efficacy. However, when the non-targeting controls were tested, the conjugate on the far left exhibited a significant amount of target-independent activity, and the conjugate on the far right exhibited the least. This was also reflected in the tolerability studies, and the conjugate on the far left showed the greatest amount of loss of body weight, while the conjugate on the far right showed the least. So presumably, this reduction in hydrophobicity of conjugate 37 relative to the other conjugates minimizes antigen-independent uptake by reducing this nonspecific uptake of the hydrophobic conjugate. So this is a good example showing how these hydrophilic orthogonal shielding PEGs can minimize this antigen-independent activity by minimizing nonspecific uptake of the conjugates. This last one is XMT 2056, which is currently in clinical trials with a different immunostimulant payload. So we'll wrap this up with some examples from the vector linker portfolio that you could use to synthesize conjugates such as these, or screen different conjugation strategies or linker variables if that's desired. So most of this webinar focused on conjugation to hinge cysteines because there's more precedent for the PEG, but site-specific conjugation strategies are rapidly becoming the dominant approach, and they also incorporate PEG components. The example on the top left is Synaffix's technology for QuickChem at modified glycans. This is ADC Therapeutics phase 1 candidate. The example on the top right is Ambrx's technology for oxime ligations at engineered residues. This linker payload is called amberstatin AS269, and it is currently in 3 different clinical candidates. The example on the left is Sutro's technology for QuickChem at engineered glycans, and this is a former phase 1 candidate of theirs. The example on the middle right is a structure disclosed in Profound Bio's patent application, and this is also for QuickChem. And the examples on the bottom are for enzymatic ligations. The one on the left is from a preclinical study by Pfizer, and this is for transglutaminase-mediated ligation. And the example in the middle is from Gene Quantum's patent application for ISACs, and this one is for sortase-mediated ligation. And the example on the right is from patents at Talec's patent application on antibody oligo conjugates. And in this case, they use a short PEG to put DBCO on the oligo, and then they use longer amino PEG azides that are conjugated to the antibody via transglutaminase, and then they link those two via click chemistry. So PEG components are certainly finding a place in site-specific conjugation technologies, and oftentimes it's just to facilitate the conjugation, whether it's to offset the hydrophobicity of DBCO or give the enzymes a little more room to work. You do have to be able to make these things in the first place. If you go to conferences or investor presentations, you'll see that these hydrophilic components in the linker are increasingly regarded as trade secrets. and they're not disclosed in these widely disseminated publications. But if you spend some time digging through associated patents, you can make some good guesses, and oftentimes they do incorporate a PEG. These linker payload designs with PEGs are also becoming more common in patents with other payload types. For instance, with maytansinoids, topo-1 inhibitors, there's antibody oligoconjugates, immunostimulants. PEG-containing crosslinkers aren't just for cytotoxins anymore. And Vector has a number of crosslinkers that you could use for whatever type of cytotoxin or payload you're conjugating to your antibody. Vector has two product lines. There's building blocks, and then there's ready-to-use crosslinkers. The DPEG-based building blocks are for you to build your own crosslinkers with your multi-step synthesis, and this is if you have some sort of proprietary reactive group or cleavable trigger that we don't offer. And yeah, you could just build it that way. We also have ready-to-use crosslinkers, and these can help expedite the synthesis of your conjugate. And this is if you don't want to mess with the synthesis of the crosslinker and rather focus resources elsewhere. For instance, the SAR of the cytotoxin, or developing the release mechanism, or maybe developing the antibody itself. These can also be particularly useful if you're looking to screen different conjugation strategies or possibly screen different linker variables. And again, you just don't want to mess with the chemistry of the different cross-linker combinations. So these are examples of Vector's building blocks that you can use to expedite the synthesis of your crosslinkers with your proprietary reactive groups and property-modifying functional groups. Now, we didn't cover chemistry in this presentation, but if you would like to go back and review these slides, and more importantly, the references and the associated supporting information, you can see how these building blocks were used to make the linker payloads in those examples. These are examples of Vector's ready-to-use crosslinkers if you don't want to mess with their synthesis. And these can allow you to expedite the synthesis of your conjugates. So they're available with a wide variety of reactive groups. One of them is typically an active ester for your amine-containing payloads. And for PEGs, we're generally looking at PEG 4, 8, 12, and 24, although others are available. For conjugating to the antibodies, we have reactive groups for cysteine conjugations, oxime ligations, disulfide rebridging, and click chemistry. The full color structures are catalog items available in high purity at kilo scale. The transparent structures have been made, but they're not kept in inventory, so you'd have to contact the sales team for details. And again, if you'd like to go back and review these slides, you can see the benefits of using similar types of linkers in some of those examples. This matrix shows the different combinations of reactive groups available on heterobifunctional crosslinkers. And this is if you're interested in something besides that active ester. The numbers in the slides, or the numbers in this table, denote the slides that use similar types of linkers in those examples, if you'd like to go back and see how they're used. The structures denoted are coded purple are standard catalog items available in high purity at kilo scale. The products coded blue are not kept in inventory, so you can contact the sales team for details. These are some examples of Vector's ready-to-use sidewinder crosslinkers, and these have that orthogonal PEG shielding structure. These are also available with different reactive groups, but one of them is typically the active ester for amine-containing payloads. For the PEG lengths, we're looking at PEG 4 for the stretcher component and 12 or 24 for the modifier. And they're typically built on a lysine or glutamic acid core, and the catalog products, the full color structures, are available for cysteine conjugations, while the transparent structures are currently in development. And again, if you would like to go back and look at the slides denoted here, you can look at some of the benefits of using similar types of linkers in those examples. This matrix shows the typical reactive group PEG termini combos on some of these sidewinders, and this is with the reactive group as the TFP ester. The numbers, again, they denote the slides if you would like to go back and see the advantages of using some of these types of linkers in those conjugates. The product coded purple is the only standard catalog item, but it is the most common malleimide active ester combination with the shielding MPEG. And the products coded blue, we have the building blocks around, but they're just made to order, so you would need to contact the sales team. Finally, this is just a teaser on some multi-payload options that we have. This is probably next-gen stuff, particularly given the rise of the site-specific conjugation strategies that might have inherent limitations on the available number of conjugation sites. The products on top are the building blocks that you can put your own reactive groups on. The products in the middle are the ready-to-use crosslinkers, and the product on the bottom is a potential custom crosslinker for a dual MOA ADC. The chemists in the audience will recognize that there's some orthogonal protection, deprotection steps. So it takes a minute or two longer, but this is certainly doable. So to conclude, off-target toxicity is one of the major obstacles to developing a clinically successful ADC. While it remains really difficult to predict the final safety profile and therapeutic window, there are a variety of preclinical assays that can be used to assess these potential downstream tox risks. And strategic linker payload design, as demonstrated in here, has a track record for improving ADC performance in these assays. Many of the underlying mechanisms for tox are related to instability and nonspecific uptake, and they can be influenced by the physical chemical properties of the conjugate and of the linker payload. Vector's DPEG-based building blocks and ready-to-use cross-linkers have a variety of peg lengths, reactive groups, functional groups, and architectures to optimize those properties and simplify your synthesis of a de-risk conjugate. So with that, I'd like to thank you for attending. I'm done talking. I know this was dense, so if you'd like, you could go back and review it, particularly read the associated references. There's a lot of people doing good work, a lot of really good work. And a little birdie once told me that we can see further by standing on the shoulders of giants. And that's certainly the case here. And if you have any other questions or information, you can visit vectorlabs.com/biodesign and look at what we have posted there. And again, thank you for your attention.
Thank you, Matthew, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the lower left corner of your screen, and we'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question asks, does the addition of the orthogonal pegs impact the yields of the conjugated antibodies? If so, does it enhance the yields of the amount of monomeric ADC?
Yeah, that's a good question. Certainly, one of the things that impacts the yields is the reactive group primarily, but many reactive groups such as DBCO are hydrophobic, so even putting a linear peg in there can help reduce aggregation and improve yields and also give it some space to work. It's also true of enzymatic ligations, where just a linear peg can help give it some room to work and improve yields. But that orthogonal peg is very helpful. It depends on the nature of your cytotoxin. If you have a payload, a trigger cytotoxin combo that you've been able to tweak its physical chemical properties, it might not be quite as helpful. But if you have some that are just hydrophobic and maybe not the friendliest to the aqueous environment, that orthogonal peg certainly helps improve yields. For instance, the tubulus example, EXA couldn't be used as a payload because it resulted in so much aggregation. And they were able to use that orthogonal peg to give them very high yields with very low aggregate. So it does depend on the nature of your payload, but in general, it does seem to reduce the amount of aggregation and improve the yields for the ADCs. Yep, good question.
Great, thank you. All right, our next question here. We have a couple of questions that actually are floating around the same topic. So I'm going to ask you a few questions, and then you can go ahead and dissect and respond. All of the cross-linkers shown as cataloged items are non-cleavable. Do you offer cleavable linkers? It appears most of the cross-linkers are not kept in inventory. Why is this? And lastly, there's always maleimide unit used for linking to antibody. Is there no other chemistry currently explored on this conjugation?
Yeah, so we can kind of amalgamate those two. I forgot the slide number, but I did address most of this presentation did focus on maleimides just because there was, more precedent with the PEG. But for sure, a lot of other conjugation chemistries are up and coming. And although we did focus on the MAL, look, the MAL NH esters, they have been the legacy workhorses for bioconjugation forever. And they're still our most popular sellers. We sell a lot of them. So we have a pretty lean group here, and we tend to focus on things we have time for or as orders dictate. But we are certainly aware of all these other conjugation technologies. We're pretty familiar with the field of bioconjugation. So we do work them in as we have time for. All these bioorthogonal reactions and the enzymatic ligations and the developments in glycan remodeling and genetic code expansion, different bioorthogonal conjugation chemistries are very popular right now. So we are working them in as we have time. And so we've already got the DBCO. That one is present as both the TFP ester and the maleimide. I think for most peg lengths, some of the other ones like the bisulfone for disulfide rebridging, we've got that as I think the NHS ester, but only at the shorter peg length right now. And then we've got a couple of the, I think we have a polygly in there. And I think TCO and tetrazine are also available as the shorter pegs. So yeah, we definitely have other chemistries to get the conjugation done, and we just work them in as we have time or as orders permit. So if you look at a lot of our cross-linkers that we see are not kept in inventory, most of the ones are those when we have the building blocks, and we're just slowly building them into inventory as we have time.
Okay, wonderful. Thank you so much for that. We're going to move on to our next one. This next question asks, how do you determine the purity of your cross-linkers, and what is your typical purity spec?
Yeah, so we use typical organic chemistry methods for determining purity and identity. This is your, LCMS, NMR, and HPLC. Those are all of our kind of typical methods. These cross-linkers do have some unique properties. Very large molecular weights, not always a chromophore present, really similar physical properties. So a lot of times what we'll do is we use orthogonal methods to determine purity. For instance, quantitative NMR and HPLC. And the legacy detection method for HPLC has been ELSD because you don't need the chromophore, but we're slowly transitioning over to CAD, which has a better response. Our purity spec is generally 95 to 98%, and this depends on the product, and it depends on your need, and on the nature of the impurity. That spec refers to like gross impurities, for instance, chemical impurities or PEG homologs. For instance, you're not going to have a hydrolyzed maleimic acid present in the maleim products, or you're not going to have a PEG 24 present in a PEG 20, something like that, right? We are getting more and more requests for the +1, -1 homologs to be included in that spec. These are homologs with one more or less ethylene oxide unit. And these come as like basically an intrinsic byproduct of these stepwise production processes of PEGs. And, that the request for that level of purity kind of depends on your application, right? One or more ethylene oxide units might not affect your structure-function relationships, but maybe it does affect your analytics, or it affects your regulatory requirements. So we're getting more requests for that, and we can do it, but it's a much more intensive process. Usually it's the +1, -1 are there in sub 1% levels. So they're almost non-existent. But if it is an issue, we can get rid of them. It's just intensive. So you'd have to contact the sales team. Anything's possible. Prices vary on that one.
Okay, great. Thank you. Our next question, moving right along, asks, how do we measure DAR?
Yeah, so the drug antibody ratio, yikes. So there's a lot of ways you can do this. Mass spec is going to be one of the best. It's going to depend on if you have if you're doing stochastic conjugation or like site-specific conjugate. If you have a defined DAR, you can usually mass spec, but if you're doing something where you're going to have a mixture of DAR species, which is the case with a lot of stochastic conjugations, then you're going to need something like hydrophobic interaction chromatography to separate the different DARs, and then you're going to have to use your peak areas to figure out what your average DAR is. So it's going to depend on the type of conjugation chemistry you're using and what you expect, but it's probably going to be something like HIC and mass spec. Unless you've got a crazy fluorophore and you can do an old school by, or not fluorophore, chromophore, and you can somehow do it old school by UV, but you're probably going to have to do mass spec or some sort of HIC method.
Got it. Thank you. All right. Our next question here asks, can you comment on any differences in administration times or methods with the various ADC designs?
Ooh, Well, I mean, I can comment. That's going to be more for like the clinicians. I'm more on the lab side, trying to get these things made and assayed. But a lot of times that the different dosing schedules are going to depend on your cytotoxin, your target, your payload, your tumor. I think a lot. I might be wrong here, but I think a lot in the clinic are dosed Q3W once every 3 weeks. And when you're doing preclinical studies, your dosing is to depend on what you're trying to show, right? So you can dose at subcurative doses just to get a general idea of how things compare at a specific dose. You're generally it's gonna depend on tox, right? If you see basically pretty intense and irreversible changes in like hematology measures or tox, then you're gonna have to slow down that dosing schedule so those don't build on each other. So it's going to depend on a lot of things and what you're trying to show. And that's about all I can really comment on that one. So I hope that helped.
Thank you. All right, next question here. We have time for just a couple more, so we'll squeeze these two in. This one asks, did Vector Labs participate in developing the ADCs shown in the slides, or are they just representative of the chemistry?
The latter. Yeah, right. So this was a literature summary of ADCs that people have developed using linkers like those that we make. Usually we sell the linker and then they develop the ADC. So it's kind of representative of the types of things that can be made with the linkers that we sell, and then the types of advantages you can get from using those linkers. So yeah, it would be the latter of those. We're just trying to provide a summary of all the things that people can find with these, and why you might want to screen different types of products that we offer, because it does depend on the type of ADC you're developing, and we can provide linkers that can help you screen some of those things a little bit quicker.
Okay, great. And then our last question that we have time here for today says, so it seems an orthogonal or parallel peg generally confers more stability versus a linear peg. Are you aware of any specific examples of the opposite, i.e. linear peg more stable than orthogonal peg? And if so, what other factors may be at play?
Well, define stability. So what are we talking linker-payload deconjugation? Are we talking payload release? Are we talking physical-chemical stability, like just aggregation? So let's see. With respect to linker-payload deconjugation, that's going to be more a function of the conjugation chemistry and not so much whether it's a linear or orthogonal peg. That won't matter as much. For payload release, premature payload release, I can't think of any. Well, there are linear cases where you can shorten up that linear peg to leverage steric hindrance from the antibody, right? You saw this a lot with carboxylesterase 1C. It also does slow down cleavage by cathepsin D a little bit, not much, and that's where the orthogonal peg that can help prevent premature payload release and improve stability by restricting enzyme access a little bit to that payload. So when it comes to premature payload release, it's probably going to be your orthogonal. And then if we're talking physicochemical stability, which is aggregation, can't think of any linears off the top of my head. For sure, the orthogonal peg does improve aggregation profiles. It can make it worse though too, right? This is why you're going to have to optimize that peg. There's some cases where maybe the peg 12, just even in an orthogonal position, doesn't help improve the physical stability, but a peg 24 might. And the big factor there is going to be the nature of your payload. If you can make a payload that is friendlier, then it's not going to require the orthogonal peg as much. But if you have a really nasty payload, like Valcit MMAE, then the orthogonal payload does help with the physical stability of it.
Excellent. And it looks like they did just pop in with a clarification. They were referring to deconjugation.
Oh, okay. Yeah. So linker payload deconjugation. Now, I think that's going to definitely be much more a function of the conjugation chemistry you're using. You know, malleimids can deconjugate, but if you're using some of these site-specific technologies, or even a lot of the neat self-stabilizing malleimids that are out there now, or the aero malleimids, those will not deconjugate. But that's much more a function of the conjugation chemistry that you're using, and less a function of the linear versus orthogonal peg.
Okay, wonderful. That really is all the questions that we have time for today. So I want to thank you for your time today, Matthew, and your important research. We would also like to thank LabRoots and our sponsor, Vector Laboratories, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.